It is the magic hour of 7 p.m. and my iPad here is reminding me that this is the time when our Zoom meeting is to begin. My name is Candace Savage, and it's my great pleasure to welcome you to tonight's presentation, our Nature City conversation, on behalf of Wild About Saskatoon. I'm going to be assisted tonight by co-host Ahmed Badawi, who will help make sure I don't forget anything crucial and step in if I seem to be losing the plot. We're using a standard Zoom meeting, so if you need captions, if you'd like captions, you should be able to find them under some tab or other on your screen. On my screen, it's at the far right at the bottom. We all are so used to this now, but I just thought it might be worth mentioning that if you use speaker view, you will have perhaps a more enjoyable experience than the gallery view. Our format tonight's very straightforward. We'll have our presentation first. If you're not yet muted, please mute yourself, else I will have to mute you, and that seems so rude. So we'll have our presentation first, followed by a question and answer with our special guest, and we'll be using the chat feature for the Q&A. So please feel free to post questions or comments there at any time. So I'm speaking to you tonight from my home near the banks of the South Saskatchewan River in Missaskwatumaniik on Treaty 6 territory and the homeland of the Métis Nation. We reaffirm our relations, which we're bound to by our ancestors through treaty, to steward this land and seek mutual benefit for as long as the grass grows, the sun shines, and that river flows. We've taken to heart the teaching of the Truth and Reconciliation Commission that justice cannot be restored to relationships among people unless our actions also include healing for the land. So tonight we are going to hear a presentation entitled Pesticides and Prairie Wetlands, a battle for the burden of proof, with our distinguished guest, Dr. Christy Morrissey. Christy is a professor in the Department of Biology at the University of Saskatchewan. Her research focuses on issues related to agricultural pesticides and other chemicals, and uses birds and insects as indicators of ecosystem health. She has published over 90 highly cited journal articles and book chapters, is recognized by the Royal Society of Canada, and is a science advisor to Canada's Pest Management Regulatory Agency. Her work has also been featured broadly in the national and international media, including the feature documentary from just a few years ago, The Messenger. I also happen to know that if you are looking for a horse and you need someone to hold your hand and help you find just the right one, that Christy is a really great person to know. So with no further ado, it is my pleasure to introduce Christy, and it's all yours.
Thank you so much, Candace, for the lovely introduction. And I wasn't expecting that about the horses. So these two parts of my life don't often seem to intersect. But I really appreciate it. And the most beautiful land acknowledgement I think I've ever heard. So thank you. All right, so I appreciate everyone for joining this evening. I'm delighted to be here to talk about something that has been sort of a lifelong, I guess, passion and interest, but also has become a current event, I suppose, in the media. So I thought I would start by letting people know who haven't seen it. This I think I was invited initially to talk about wetlands, but also when Candace asked me to do this presentation, this was right around the time when in last October when I was in the media for a story in the National Observer that was how a federal agency colluded with a pesticide maker to silence a Canadian researcher. And this kicked off quite a storm. Of course, Health Canada started to investigate this, and there's been further articles even as recently as this January. Basically, the controversy continues as to whether or not the federal government and Health Canada have been colluding with industry. And I'm really here to tell you a little story, essentially, about this. And for those of you who care about prairie wetlands, hopefully I can impart why I'm so passionate about this issue. But the story really starts with pesticides, and not all of you maybe are aware of the pesticide industry and how agriculture works, and sort of the evolution of our increasing use of pesticides in agriculture. Obviously, most people have heard of Rachel Carson's DDT and the DDT era and sort of the damage that was done by DDT and related organochlorines back in the 1940s when those were released. This was considered miraculous. Indeed, Mueller received a Nobel Prize for inventing DDT. So this particular application was, of course, a milestone in science and in controlling pests, and certainly pesticides have done a lot of good things in terms of allowing us to expand our acres of crops grown, the types of crops grown in different climates and environments. But there's also been a backstory throughout the ages of different chemicals that have been released onto the market and their potential hazards for things that they're not intended to kill, that are not pests, including birds and wildlife. Now, there actually has not been very many insecticide products that have come out over the years that are in different classes. Many of them that come out are very similar in how they work on target insects and non-target pests, non-target pests. And the neonicotinoids came out in the 1990s, basically as a revolutionary group of chemicals. Because they were designed to be primarily used as seed treatments. So this meant that farmers could apply them to the coating, or they would buy them as a coated seed, and that provided plant protection. Now, there are a number of different neonicotinoids registered for use, and I've listed some of them here. The controversy, though, about these chemicals, although they're kind of miracle chemicals in that they are very effective at killing a bunch of pests, they also tend to contaminate entire ecosystems. The neonicotinoids are chemicals, as I said, that are applied to the coating of seeds often. And the problem is not all of the chemical goes into the plant. Much of it is actually retained in the soil. It's estimated between 80 and 98% stays in the soil rather than being taken up by the plant. And indeed, that doesn't always stay put in the soil. It can leach into groundwater or run off into surface waters. When it contaminates surface waters, then that can deplete aquatic invertebrates to which birds and other wildlife are dependent on. And it also can affect the plant, and there are residues detectable in the plant's nectar and pollen, which can contaminate non-target beneficial insects like bees and butterflies. Now, the neonicotinoids are produced principally by two major companies, Bayer Crop Science and Syngenta. They came out with the first generation of neonicotinoids, which was the imidacloprid, and then the second generation includes things like clothianidin and thiamethoxam. These chemicals are very widely used around the world. In fact, they're sold in 120 countries worldwide. They've been registered on over 140 different crops. And it's estimated that 80% of the global market share in seed treatments contain neonicotinoids. And the total market share for all insecticides is about 30%. So this is an important chemical to the chemical industry, to the agrochemical industry. It is an important chemical class for farming and farmers, and indeed, it is used here in Canada and most of the world. So they're controversial, though, because not only are they used everywhere, but they persist in the environment. They move. They don't stay put on the seed or indeed right where they're applied. And they are extremely toxic to things that are pests, but also non-pests, including beneficial species like bees, beneficial insects, as well as birds and aquatic invertebrates, and even mammals. The focus of today's presentation is on wetlands, and I want to start by saying that neonicotinoids and water don't mix. They do move into water, as I mentioned, and this is because the properties of these chemicals are designed to be seed treatments that are taken up by the plant's natural xylem and phloem. So they're actually a systemic pesticide. They're designed to be taken up by the plant and give it protection from biting and sucking insects. And as a result, that means that they're made to be very water soluble. So when they're water soluble, they will move very easily off of the seed or out of the soil and be washed out and end up in places that they shouldn't, like including wetlands. Their solubility is actually so high, it's sort of like salt and water. For reference, I put here thiamethoxam, one of the most widely used neonicotinoid insecticides, has a solubility of about 4,100 milligrams per liter. Most other insecticides that are commonly used, including chlorpyrifos, which was recently banned, has a solubility of only about 1.4 milligrams per liter. So most of the chemicals that are out there don't end up in water. And if they do end up in water, they typically are broken down very quickly, often within hours. And in the case of neonicotinoids, they don't break down very easily in water and require light to be degraded. Here in the prairies, we've done a lot of water sampling to test whether or not these chemicals are detected in our surface waters, mostly wetlands, because those are what we have primarily as our surface waters that are of ecological relevance. And we found that between 16 and 91% of the wetlands that we've sampled, depending on the season, contain at least one neonicotinoid. And over half had more than one neonicotinoid. And the concentrations were in the ranges that were well above toxicity thresholds that would affect aquatic insects. The interesting thing is that most detections that we see of these chemicals happen before farmers even put seed in the ground. So this means that a lot of the chemical actually sticks around from the year before and stays in the soils or in these wetlands all through the winter, and we can detect them in the spring in the snow melt and indeed in wetlands. They are detected in other places around the world very routinely. Scientists have been studying this now for several years. In the US, in a widespread water monitoring program, they had 38 nationwide streams that were sampled, and virtually all of them have neonicotinoids in them, and often more than one of the neonicotinoids. So here you can see on this graph different neonicotinoids: imidacloprid, clothianidin, thiamethoxam, dianetrofuran, and acetamiprid. These are all neonicotinoids, each one of them fairly commonly being detected in this case, river samples. They're also found in drinking water. This was a study done in Iowa, and they found routinely 3 different neonicotinoids in both source water and in the finished drinking water after it had been treated. They've also found it very frequently in wastewater treatment plants, which means that it's going through various wastewater treatment systems, and across the US, there was 100% detection in effluent of imidacloprid. So where are we with all this science? It's been, I think when I started studying neonicotinoids in about 2010 or 2011, there was very little out there. And, most people didn't know what these chemicals were. But over the last 10 to 15 years, the number of documents published or the number of papers in scientific journals and elsewhere have skyrocketed. There's so much science now that we actually, when I did a Web of Science or a Scopus search for this, just in preparation for this talk, I found that there were almost 33,000 documents published in the scientific literature on neonicotinoids. So that is a huge number. And it turns out that in 2012, which is where you can kind of see on the graph things starting to take off, this is when Canadian and US regulators just basically made the discovery that these chemicals are potentially controversial and that they contaminate ecosystems. And that's when, things really caught on. So much of the research has happened since 2012, even though they've been registered for use since the early 90s. And I'm going to talk mostly about this issue around when neonicotinoids were essentially banned and what happened in this period when there was a lot of science going on. So keep in mind this 2016 to 2021 period. It turns out that the regulators in both Canada and the US and in Europe have been doing a lot of research and a lot of work to try and understand whether or not these chemicals are so-called safe. In 2016, the Health Canada, after their 4-year review, decided that neonicotinoid, including imidacloprid, should be banned. It was the risk assessment showed that it was harmful. In 2018, the European Union also implemented a full neonicotinoid ban. In 2018, Health Canada decided that not just imidacloprid, but all the neonicotinoids should be phased out in the next 3 to 5 years. By this point, everyone I know was celebrating. In 2020, US EPA followed suit, but they were still reevaluating neonicotinoids. They continued to register it, and so this was a bit curious given that other parts of the world were already on track to ban it. And in May 2021, there was a bit of a surprise because Health Canada decided to reverse their proposed decision to ban or phase out the neonicotinoids. And this was perplexing because all signs were pointing that these chemicals were harmful, both in the scientific community and in the regulators around the world. So this prompted me to wonder what was going on and why did they reverse the ban? This news was, I think, somewhat shocking to the scientific community, given all the evidence we had been collecting, but also because PMRA, in their original decision, their proposed decisions for imidacloprid and the other neonicotinoids, decided that these were harmful to aquatic life. In fact, they wrote this in the proposed decision of 2016 and 2018, that the environmental assessment showed that in aquatic environments in Canada, imidacloprid is being measured at levels that are harmful to aquatic insects. And they recommended canceling all outdoor agricultural, ornamental, turf, and tree uses, except tree injection uses and greenhouse uses. So this was a pretty strong message that these chemicals were not safe for aquatic ecosystems. The final decision, though, in 2021 was a flip-flop. And now this was the language in the document from Health Canada. risks are largely acceptable with mitigation. So mitigation that they proposed was that some outdoor uses are not acceptable and will be canceled. And additional mitigation measures like rate reduction, spray buffer zones around water bodies, label changes will be required for some of the uses. So these mitigation measures were what they decided to go with instead of presenting an all-out ban. So in 2021, after that came out, I filed a notice of objection, which is what PMRA, the Pest Management Regulatory Agency of Health Canada, has as the only option to sort of object to a decision on any chemical registration. So in July 18th, you can see a copy of my letter. I wrote to the Minister of Health to submit and file a notice of objection. Now, for those of you who have never done this, your notice of objection must be submitted within 60 days of any decision from Health Canada. So I filed the decision within the 60 days. However, I had also requested that there was data that I'd like to see to inform my full notice of objection to better understand how they came to this decision to reverse the ban. It took them 9 months to give me the data, which I had to receive through a secret reading room request, where you get sent an encrypted data file on a thumb drive. And on that data file, you also, or to receive the data file, you also have to, sign a notarized agreement to not share it. And indeed, it took a long time to get this. Once I received this encrypted thumb drive, which contained some 170 different files that were unnamed, and many of them hundreds of pages, or PDFs of documents and Excel files, I then compiled all of the information and submitted a full notice of objection with additional evidence in October 2022. I can tell you at this point, I still have not received a decision on this notice of objection. I'm still waiting. So I can tell you a little bit about why I think the PMRA changed their decision between the sort of 2016, 2018 decision and the 2021. And I believe that it was because they were bending some of the science that was out there. There's two areas that I presented in my notice of objection about effects on aquatic invertebrates, and also some of the mistakes or errors or assumptions that they made on its safety to birds and mammals. I'm not going to talk about the birds and mammals here. We'll have to have another webinar to talk about that. But the aquatic invertebrates is the focus of today because we're interested in prairie wetlands. It's also the reason that they originally banned the chemicals, was on the safety concerns for aquatic life. So I'll tell you a little bit about the aquatic invertebrates. I recognize this is a bit science heavy or science technical, but I'll try to walk you through this so that it's easy to understand. The risk to aquatic invertebrates, basically the decision was because of issues around chronic toxicity, that is organisms being exposed for longer periods of time. In most toxicology studies, we characterize acute toxicity as any kind of test that is done where the animal is exposed for less than 96 hours or chronically exposed for more than that. And usually it's quite a bit more than that. In the case of aquatic invertebrates, it's usually something in the range of 14 to 28 days, sometimes longer. They also looked at the Canada water monitoring data, and that included all data on what the levels were found in water bodies across Canada. There's a lot of information, a lot of numbers on this table, but all I want you to get out of this is that the concentrations that are safe to aquatic life of these chemicals has been reviewed by agencies around the world and academics like myself. And we've all come up with a number of what is the safe level. And some of the older data wasn't as complete, and the concentrations that they felt were safe in terms of a reference value were higher. But in more recent evaluations, with the exception of the US EPA, which everyone acknowledged was wrong or incomplete, all the other data from Canada and Europe, the Netherlands, as well as studies done by myself and colleagues, all pointed to these very low levels that were safe. So a level of about 0.035 micrograms per liter is safe for organisms that are exposed chronically, or 0.2 micrograms per liter would be considered safe if they were exposed acutely for short periods of time. Now, the PMRA, it turns out, changed. Well, I guess in 2016, the concentrations they thought were safe were very similar to what I came up with in 2015. But by 2021, this number went up by, in some cases, an order of magnitude. So it increased like 10 times higher. Why does that matter? I'm going to show you why that matters. But first, you need to understand how these numbers are derived. These numbers are derived by compiling toxicity data from all kinds of toxicity tests that are done in the lab on various species of aquatic insects and aquatic crustaceans. You don't need to know what all of these little critters are, but if you are interested, the one here at the top of this curve, Daphnia magna, is the water flea. Many people will recognize it. This particular species has been used in toxicity testing for decades, and it is commonly considered a sensitive species to a lot of different pesticides. This was the species that the industry used to submit their toxicity data to the Health Canada and other regulatory agencies around the world. But as it turns out, Daphnia magna is uniquely insensitive to neonicotinoids. This curve that you're seeing on the graph is called a species sensitivity distribution, or SSD, and it's showing the ones that are most sensitive are down at the bottom, and the ones that are least sensitive are up at the top. So the most sensitive species are the ones where at a lower concentration, they cause harm. Usually it's death or some kind of developmental effect. And things that are on the bottom tend to be insects, usually things like midges, sometimes it's mayflies and black flies and other things. But the things at the top tended to be these crustaceans like the Daphnia magna. PMRA actually went against their own advice, and they decided to not use the species sensitivity distribution. Instead, they used one value on one species called Cleon dipterum, which was derived from a single mesocosm study. And that is completely inconsistent with their usual method. It turns out that deriving from this particular method meant that their water quality safe values or reference values went up. I'm just doing a quick description here of what happens when you use different values. If you use the species sensitivity distribution, the value for imidacloprid would be 0.061. If you use the PMRA's level of concern for a mesocosm, it would be 0.16. If you do it for the other compounds, you get kind of the same result. It's about 10 times higher if you use the PMRA's new mesocosm value. And that means that the level that is safe is now suddenly 10 times higher than what would be considered safe under most risk assessments. So let's talk about the water monitoring data. This is really interesting because after the first decision came out in 2016, while industry and governments, provincial governments, and a bunch of other groups got together and decided to form what was called the Pan-Canadian Water Monitoring Network. Canada doesn't have a national water monitoring program up until the last 2 years, which was a recent addition. We have not had systematic water monitoring data. So this was an exciting initiative where various industry reps and others got together and said, we're going to actually go out and sample water bodies across the region, all regions of Canada that have agriculture, and we're going to find out whether neonicotinoids are truly exceeding these levels of concern. But the problem is, it turned out, and I sat on this committee for initially, but it was basically run by industry who collected most of the samples. They collected almost 9,000 samples from 765 sites. Most of the data that went to the PMRA under this submission were new data. And the prairies had a lot of data, it turns out. 99% of the samples and 93% of the site years were all new data that was provided after that initial decision to ban neonicotinoids. It turns out when I looked at the reading room data, most of these samples were the same water bodies, often clean sites that did not have the chemicals being used in the land around that water body, and they were resampling the ponds up to 22 different times in a season, which biased the overall data set. So it looks like you have a really big sample size of almost 5,000 samples, but most of them are from the same locations, just sampled over and over again, and usually from sites that were already considered clean and not contaminated. Interestingly, PMRA then did an analysis and decided that there was virtually no exceedances of their level of concern. So by raising this bar to a new level of 10 times higher and sampling ponds that are clean, you actually can now change the result. I dug further into this, and it turns out that they also excluded much of the published data in the literature. In fact, they looked at our data from prairie wetlands that we had sampled in 2012 and 2013 and 2014, which many of them exceeded our original threshold for toxicity. But when you use the PMRA's new value, which is 10 times higher, now they don't exceed it. And indeed, if you use the other value for clothianidin, it definitely doesn't exceed it. But it doesn't even matter because PMRA got dumped all of this data and they didn't use any of it, citing that the reason was the land location data were not provided. So they actually didn't use any published data in the second evaluation. And we had done a lot of work in one summer in 2014, and I had shared some data with PMRA, that additional data that was unpublished with land locations. And we had found a number of them did exceed that threshold level that we determined was safe, but they didn't all exceed the ones that the PMRA decided was a safe level. But interestingly, in the final decision document, most of these samples were also removed from the risk assessment. And indeed, they cited that the reason was that they were not relevant, that the sites chosen were not relevant. So this had me scratching my head as to why this data was robust and used in the initial assessment. It was published data or very robust analyses, but suddenly these were not relevant, or land locations, which is revealing a landowner's where a landowner was and where the pond was suddenly now criteria for getting rid of the data. And I figured out after digging through one of the 170 documents that came in the reading room that it was because this is what there was a document in there called Relevancy of Monitoring Sites for Aquatic Risk Assessment, a classification of 2014 water sampling locations. Brackets, something, but I know it said Morrissey because so they actually redacted the name Morrissey throughout the entire report. But it's based on our data. They went out and hired a team of students or scientists to go out and take pictures of the sites that we had been to 3 years later and during a drought and during August. And they determined that many of the sites were dry. So the wetlands that we had chosen to sample had water in them, obviously, but they just went out a few years later and said, oh, these sites aren't relevant because they're dry now. And as a result, that was the reason why PMRA said that they were not relevant. So I think there's a lot of evidence now that's building that the regulators are being influenced heavily by industry. Most of the data that they use is not in the published literature. I'm showing you here that out of all those thousands of studies or documents that you can find on neonicotinoids, only 2% are in the published literature is funded by Syngenta or Bayer as the authors and the funding organizations. Most of it you have to go through this so-called reading room, which is very, very hard to do. And of course, this is coupled with the power struggle of having a seed industry that now runs and controls the chemical industry that now runs and controls all of our seeds that are sold around the world. From 1996 to 2008, this is a figure showing the big six, which are the six top agrochemical companies in the world: Monsanto, DuPont, Syngenta, Bayer, Dow, and BASF were the top six producers of agrochemicals and also own most of the seed patents. By 2018, the picture changes a bit because Bayer has now consolidated, for example, and taken over Monsanto. Among other changes, like ChemChina has taken over Syngenta. And so now we have the big four. These big four firms now control an estimated 60% of the global seed sales. And that means that all of our food that's produced is produced with the control of these organizations that are basically running a lot of the agricultural industry. So I think the pesticide regulatory process has a number of issues. There's delays, there's a number of biases, and I think industry influence of an ever powerful, in this case, big four, are certainly having a large impact on controlling the decisions of our pesticide registration. In the particular case of the neonicotinoids, they didn't use scientifically accepted methods for deriving water quality guidelines. They chose to use mesocosm data instead of their own guidance and using species sensitivity distributions. They relied heavily on registrant-provided data and reports on water monitoring. There was long delays between the decisions. This allowed industry to go out and collect their own so-called science in the interim and flood the data set. They excluded or limited their use of published peer-reviewed data that's coming from independent scientists like myself. They also have a lack of consideration for all the effects that can occur. For example, it's for species at risk. And indeed, there's been new research showing that endangered species in the US are heavily affected by these chemicals. And then on a general point, I think the influence or the direct weight that industry has on the decision-making of our regulators is too strong, and that there's a lack of transparency and trust for the public in understanding how the data is used in risk assessment. It's very hard to see the data they use. It's kind of secret. And indeed, my notice of objection is still being reviewed some 3 years later with no response. So I'll just end by saying that the motivation for me to keep fighting this fight is because I believe wetlands are important for prairie ecosystems. We have to protect them, and that they're increasingly under threat from agrochemicals that are inadvertently moving into these systems, which can harm aquatic life as well as the birds and other critters that depend on them. Thank you very much for listening, and I'm open to hearing questions or just a chat or comments about what I talked about today. Thank you.
Christy, where do we begin? There are very important questions in the chat, and many very well-informed comments. So I refer people there. I wanted to ask you a question about, well, in the first place, really, are we in the situation where to feed ourselves, we have to poison our entire surroundings? This is really, it's really so appalling to think of fields of poison plants and contaminated water bodies everywhere. Just very, and so much corporate control. I'd like to know, in Europe, for some time now, some neonicotinoids have been banned. And what is the consequence of that? Has agriculture simply shifted to other products? Is it an improvement?
Yeah, it's a great question. I mean, I think everyone's been watching what happened after the European Union ban. First of all, did agriculture collapse? No. So that was the gloom and, doom and gloom scenario. If you take these chemicals away, indeed, everything's going to collapse. So there has been a shift to other chemicals. I will tell you that just banning chemicals is not the solution, Candace. It's because there are new chemicals that have been released onto the market that are just as bad or worse than these ones. And they are increasingly being found in our wetlands, and I've been sampling for those. So although, I'm passionate about this issue, about this ban, that this was an error. I truly believe this was an error on the regulator. I also think that fighting that fight alone is not the correct one, and that we should be looking at other ways to grow food with less chemical. I'm never going to say that we don't need it. There is always a potential use for that tool, but we do need to look at alternatives as well as agricultural practices as a holistic system that needs to be revised. And I am doing a lot of science now on solutions, which are, sustainable agricultural practices, regenerative agriculture, taking marginal land out of production and trying to plant perennial forages to reduce our agrochemical use, add diversity to these agricultural systems, and indeed giving producers the tools and the knowledge that they need to scout for their pests in their fields, rather than applying prophylactically. But great question.
So we just received news this week that the Federal Court of Canada has ordered Health Canada to review its findings about glyphosate,
Right.
because they did not consider the most up-to-date research. So glyphosate being the most commonly used herbicide
Yeah.
in Roundup and so on. And there's someone in the chat here
Excellent. Yeah.
mentioning also interacts with neonics in some bad ways. So is there a possibility of using the courts to have the restrictions on neonicotinoids?
It is the only option left, and I am exploring the use of the courts. Correct. So I can't go into that more, but at the moment, because they aren't the Pest Control Products Act says that your only mechanism is to use the notice of objection. I've already done that, but if they don't answer your notice of objection for 3 years, then there is no recourse for any other mechanism, right? So there's media, and that has shaken things up, and it did go through the House of Commons, and there was a stir, and they are looking into it, but it still hasn't produced an outcome. They tell me they're working on it, but that still isn't an answer. So the courts are increasingly becoming the source for correcting decisions that are made by PMRA or indecisions. It's a shame that that's how it has to go, because I think there are transformations that need to be done within our regulatory system to make them more robust and more transparent and indeed more timely.
I see that there are several people with their hands raised here. If you would use the chat, that would be the best way for us to manage questions. I promise that I'll watch very carefully. We have a question and a comment. Has Safe Food Matters and lawyers from Eco-Justice had a chance against, or do they have a chance against these big four companies? Or do we need to go after Health Canada? And then, thanks to you and other
Yeah.
notes.
Yeah, I think both those organizations are very active in this space, not just for the neonicotinoids, but other chemicals, including glyphosate and others that are controversial. And so I know that they are active. This is an important part of our, our system that is, again, to try and correct some of these issues, because, when the government makes mistakes, or we believe they make a mistake, then, they do have to show that or prove that. I think the better solution would be if these things could be more transparent. Like the data, for example, is called confidential test data. I fail to understand why confidential test data is so top secret. If it's toxicity data, for example, it's not proprietary information that researchers like myself want to see. We just want to see what is the toxicity data, not, the chemical composition of their formulated product that has, some secret sauce in it. We just want to know about how toxic it is. But this is all confidential test data, and you have to, jump through a million hoops to find it. So I don't think that's the way to go. It's true that we just have to always go to these very extreme mechanisms to solve that. And it would be much better if we could just have the data more open, have the ability to check. And if the regulators were being checked by scientists, then they may make decisions that are perhaps better. Maybe they're the same, but maybe they're not.
We have a question from Cheryl McCumsey about the synergistic impact of neonics and glyphosate, and also the volatility. Chloropyrifos is found in all Arctic media dating back 40 years. Is it moving in the air? And she has a lot to say. She also says, we don't monitor pesticides sales very well. And she's been told that seed coatings are not included, even though we have a dramatic increase in general.
Mm-hmm.
So,
Lots to unpack there.
yes,
It's
yes.
great. Thank you, Cheryl. You obviously know your stuff. The idea of synergism between multiple chemicals, I mean, I can tell you that we have monitored wetlands for more than neonicotinoids. We monitor them for many chemicals, and we typically find, a dozen or more chemicals in wetlands in any given water bottle that we would sample during the growing season. And glyphosate is one that is in virtually every water body. We don't even sample or analyze for glyphosate anymore because it's very expensive. It's a separate analysis, but it's in everything. It's in every water sample we ever sample. So is there a synergism? Maybe. We just, the data to determine synergism requires like tests that are pretty complicated, and we never do those. Like the regulators don't do them. They regulate on single chemicals rather than on multiple products. But I can tell you that many of the chemical companies do patent formulated products that have multiple active ingredients because they are more, they act in a synergistic way, and that they are more toxic to the pest and obviously non-pest, but that's not what they're not advertising that, but they are more toxic to the pest, which means that they will be more toxic to non-target organisms as well. And in terms of your question about volatility and moving to places around the world, yes, some chemicals are important, have important properties like being more volatile, and those can end up in air and move long distances. The chemicals that were worst at that were the organochlorines. So ones that have a very high persistence profile and high volatility are flagged most of the time in our risk assessment, early risk assessments, to try and remove those. But it's true that there are many that can move much longer distances than certainly on the field. So that's a challenge. And seed coatings is the last one, but I think, yeah, the issue around seed coatings and sales of those chemicals has skyrocketed, in large part because this is an approach to agriculture that is simpler for large scale production, right? You're applying the chemical to the seed and it's easier. So that's why it's being used that way, but it has not, there is data to show that it has not reduced our reliance on other sprays, for example. We still use the same amount of the other chemicals, even though we're using seed treatments. So it has not replaced anything.
Sean Clark asks, are the impacts the same in native grasslands, grassy fence lines, native habitat, etc., that are intermixed around crop fields?
Great question. We always see that grasslands have lower concentrations of these like if wetlands are surrounded by grasslands, they have lower concentrations, but they are usually not clean. So what does that tell me? It tells me that these chemicals can move longer distances through probably through groundwater and subsurface connections, and possibly through aerial deposition, but they are not necessarily clean. So most wetlands that we sample, regardless of land use, have these chemicals in them.
Are they always at ecologically significant levels?
Not always. No, there is probably, we estimate that for a single neonicotinoid, the exceedance is varies by year, for example, and by season, but you can sort of on average, probably about 30% of the wetlands would have exceedances. In some cases, it's higher than that, but more like 50%. But nonetheless, a significant number have exceedances in any given season or year.
Depending on whose definition, who's drawing the line, right?
Well, that's also part of it, but even using our more robust one, but it is true that many of them are exceeding this, and
Right.
that's based on a single chemical. So when you start to combine them into the overall toxicity, then we'd be well above that.
Rod Olstead asks, could you describe the different formulations of neonics? I've heard of seed coatings, but are there other ways of applying them?
Yeah, great question. It is approved for use as seed treatment, as I mentioned, but also as a spray and as drenches, which are like in-furrow drenches where they just sort of fill it. Those applications for like drenches are now more restricted, but the seed treatments are really, like here in the prairies, most of it is used as seed treatments. Maybe potatoes might be the only one that's still sprayed. But in other like vegetable crops, it's mostly sprays, like blueberries and so on in the West and all that. They use a lot of imidacloprid as sprays. And there's concern for the bees and pollinators from that application. But the seed treatments are ones that are kind of like harder to see, right? Because you put it in the ground and you don't see any spraying. That's one of the advantages to the use of seed treatments, but they are not connected to a pest pressure. So they're applying it prophylactically at the beginning of the season, regardless of knowing whether or not they need it. And that, I think, is problematic.
Elizabeth Beaubien wants to know if you have enough money to do your work and do you need more students?
Oh, oh, well, I don't take any money from the chemical industry to do my research. So I'm primarily funded by either government or like NSERC, which is our Natural Sciences and Engineering Research Council. And I've been able to do the research so far, and I'm very, but I also find that it is challenging to do this kind of research. Most of the time in agriculture, most funding comes from, industry or other supporters that are wanting you to find them a solution to a problem that they're having, but not usually chemicals. So it has been a challenge, but I have managed to do that. And students are always welcome. I always looking for good students. But I do have a lab with about 10 students in it and postdocs and so on. And it's a delight. That's a great part of the research, is to work with students.
Someone else wants to know if you're available as the guest speaker and as a
Of
result.
course.
Yes. Pretty
And I
easy
think this
time.
kind of there's someone who's working on a bit of a podcast on this sort of topic, because they felt like this story, I tried to tell it to you in half an hour, but of course I left out lots of the details. There's lots more I could tell you.
Branimir has a question for you. You
Okay, go
proposed a
Branimir.
solution to reduce the use of chemicals by increasing the amount of semi or natural habitat. Can you say something about the economics of retaining such habitats, wetlands, prairie, shelter belts?
Yeah, I mean, this is an area of research that I am really passionate about now. I am fully aware that farmers need to make a living, and it is doesn't escape me that they're running small businesses. And if you have all the variation from weather events and other reasons why you might lose your crop, the thought of losing your crop to a pest is significant. And the insurance value of having a chemical is also a strong motivator to use these, right? Because you want to protect them. Now, I I am of the mindset, though, that not many growers want to take land and just like stop applying chemicals as a result. I don't really think that they're going to do it. And there are many other methods like intercropping, you're aware of, Branimir, and cover cropping, that can reduce their chemical reliance. It's great, but a lot of them don't want to do it because it's hard. So we've been trying to do work on these systems by working with farmers to map where all the marginal land is using their yield data. And land that's low profitability, low yielding is areas that we are working with them to take out of production and put in as grass, for example. Now, I have a secret to tell you, that it's most of the time the marginal land tends to be next to wetlands. When we look at their yield maps, very commonly, these areas are like red rings, which are areas that are losing money. They are low yielding. So they should not have been cropping there in the first place. And we have data that shows that if they buffer these wetlands and keep the margins intact, like all the zonal areas of wetlands intact, you can lower the chemical concentrations in the wetlands significantly to get them below these toxicity thresholds. So this is a big area that I'm working on now, is science of under what's called the Prairie Precision Sustainability Network. You can look it up, prairiepsn.ca. And we have a big group that are trying to do this across the prairies to work with farmers to convert these areas, their marginal land, into perennials. And I'm really excited about this opportunity to find a solution, not just tell them about the problem.
This leads right into Bonnie Lawrence's question. Is there any reliable information on the size of safe buffer zones, and is there a difference between tame forage versus native?
Yeah, great question. We know a little bit about forages, using forages as buffers. One of the things is the province recommends 30-meter buffers just as a standard against any riparian zone. And then other documents around in the US say 10 meters. There's no science behind that number. And indeed, it matters more about, in terms of wetlands, about maintaining the actual zones. So wetlands have kind of multiple zones, sort of from the inner zone to the sort of deep emergent marsh and the shallow marsh, and then the low prairie. Most growers are cropping right up to the cattails. That means that they are actually in the middle of the wetland. If they're cropping to the cattails, which is where their equipment gets stuck, that is called the deep emergent zone, and that is the middle of the wetland. So if we just not think about it as meters, but rather about like what plants and what sort of zones, if they can protect all of the zones, then there is that is the advice I'm trying to promote, rather than simply how many meters from the water edge. Because I don't know about you, if you've ever watched wetlands, they dry through the season and the water edge is changing. So it's not a good metric to use the water line.
Jody has a very interesting point here. She's talking about the research that shows the amazing migration of water boatmen from wetlands into the river and the way that they support fish. And she'd
Yes.
like to know, has this been used as a metric to evaluate the effects of these persistent pesticides?
Yeah, Stephen Traco did that work with Tim Jardine, and it's pretty cool. These wetlands are a source of emergent insects and emergent water boatmen, which is amazing that they can move and migrate from these wetlands to the river. We are just starting to quantify now on another project how much biomass is being exported by wetlands in terms of all insects, all invertebrates. And that we also are doing some controlled experiments to manipulate when chemicals are added, how much does that reduce the overall biomass? So that we can get estimates of what the impact is on this overall phenomenon of emergence patterns. If chemicals are contaminating 50% of our wetlands, how much would we lose in terms of that biomass? Because I know it's a big number. It's hard to think about that, but there is all this mass of insects that are coming out of these wetlands and being transported into the terrestrial environment or other places. And that's a huge part of the value of our ecosystems that we're not often thinking about when we look at the water. Great question.
I think we have time for two more questions. Aurelie McPherson is hoping that you would comment about the new water stewardship policy in Saskatchewan, which is based on the estimate that we have 86% of the original wetlands, and therefore we can afford to do a lot of drainage.
Oh, I have a lot to say on that. In short, I'll just say, in short, I don't think the policy is sufficient. And I think that we need to push back on that. And this is because it's not just about a total percentage, and it's also one of the nuances of that policy is that it's allowing consolidation or not recognizing the ones we've already lost in part. So it is not recognizing the fact that wetlands are these interspersed of all different wetland classes and sizes. And if you just consolidate them all into one big super wetland and then call it a day, it's not about total area. It's got to be about also distribution. And you also have to take into account what's historically been lost. We can't do it as a reference from today, for example, because we've already lost, say, 70%. So I'm not in favor of that policy. And I don't think that the numbers that they're using, no one that I know who's a wetland scientist also can come up with the same numbers that they have. So I'm not sure how they derived it. So again, when these policies come out, it's nice if governments make policy, but they have to show how they develop them. Like how did you make that decision and provide the data in support of it? Because that's how science works. And that's how I get my research published. I have to be scrutinized and show all the data before I can make these claims. And I think that our governments need to do this, stand to the same level of standard.
I'm going to cheat. Another question.
Okay, yeah.
Is the podcast going to be available? The podcast you mentioned?
Yeah, I would hope so. It's still being recorded. I think we've had like 4 sessions already. So 3 or 4. So yes, yes, I think that's the goal. But yeah, because there's lots more, as I said, to say on this topic. I wish I could you know, I wish I could I probably could fill a few more hours, but these are all really really good questions and really good discussion. So yeah.
And brilliant resources in the chat. So final question, how can we help?
Oh, how can we help? Write to the minister about your interest in both at the provincial and federal level. But if you're interested in pesticide regulations, that is Health Canada. And Health Canada has a division called the Pest Management Regulatory Agency, PMRA, and they are responsible for regulating all of our chemicals that, that ends up on food that you and I eat. But indeed, also, these are chemicals that are being released into the environment. And there is always concern for environmental health. And if you're concerned about this, then I encourage you to make a statement about that. And also try to think about where you buy your food and how you what kind of things you support. There are many of the industries that are now very sensitive to the fact that the public is becoming more aware of how their food is grown and indeed want to see more sustainable produced food. And there is increasingly from places like General Mills and other kind of big companies, McCain and all this, who are increasingly aware of how the growers are producing that food, and they're trying to work with growers who are using sustainable practices. So it is a valuable way to think about where you buy, just like we're doing now with buying Canadian and not American goods. You can think about where is your food coming from and buying from sources that are using more sustainable practices.
I don't think a few words could really convey how grateful we all are to you for all this work and how much we admire you for your determination and intelligence and courage in doing it all. Thank you. Thank you for this presentation, but thank you for so much more.
Oh, thank you so much, Candace, for the invitation to talk to this group. And I really enjoyed the discussion as well as looking at all these incredible chats. I'm sorry if I didn't get to everybody's comments, but you're also welcome to email me if you want to follow up with anything. I'm happy to try and respond that way. So thanks again, Candace, and everyone for listening and inviting me here today.
Thank you. Before we all go, I should just remind you that on March 19th, Wild About Saskatoon will be sponsoring the final conversation in our winter series. This one is called Indigenous Relationship with Plants, and we'll have 4 brilliant speakers. So it will be very rich and another opportunity, perhaps more joyful opportunity to learn. But thank you again. Thank you all for coming, and especially thank you and huge round of applause, Christy.
Thanks again. Have a nice evening.
Bye. Thank you so much.
Bye. Thanks, Candace, for